Good morning, everybody. It's a great pleasure to be joining you from a rather chilly morning in London. I hope that you are all well and very excited to be talking to you in this session on emerging infections today. My name is Aubrey Cunnington. I am based at Imperial College London, where I'm the head of the section of Pediatric infectious diseases and a reader in Pediatric infectious disease. And I also work as a clinical consultant at St Mary's Hospital in London. And I'm going to be giving you an overview of emerging infectious diseases for the next 45 minutes or so. So I hope you can all see my slides fine, and I hope in a second, yeah, they will start advancing. Brilliant. So I want to just explain the aims and objectives of this session first. I want to give you an overview of emerging infectious diseases, and I hope that by the end you will understand what defines an emerging infectious disease, the types of infection which emerge, and the drivers to emergence of infectious diseases. I'm going to review some recent emerging infections, no prizes for guessing what one of them may be, which have had catastrophic consequences. And I'm going to consider what can be done to limit the emergence of infectious threats. So let's have a definition first of emerging infectious disease. Actually, it's quite broad, and it can be either the recent appearance or detection of a new disease in a population, or the amplification of transmission of previously rare pathogens that increases either their incidence, their prevalence, or geographical distribution. So these are some examples from a paper which is now a little bit old, but still one of the seminal papers in the field, really. And you can see examples of some diseases that have infectious diseases that have emerged around the world, and with quite different patterns of emergence. So if we take the example of West Nile virus to start with, in the United States, previously absent and then suddenly appeared. So that would be fit one of the definitions of emergence, where it was new detection of a disease. But then you can also see next to that in the United States, Lyme disease, which has emerged, but in a different way. It's just been a steady increase in cases over the time 1980 to 2012, as in this, this graph. And then you can see, perhaps turning to something closer to home, tick-borne encephalitis, where previously there were low levels of cases, and then around the mid-1990s, there was quite a big increase in cases that was then sustained. So that's a different form of emergence again. And bringing it to something more up to date, but still not the current emerging infection, we had the example of measles, and this was talked about at EIP meetings a few years ago with presenters from several countries describing the measles epidemics happening in their countries. And you can see that there was a real problem with measles. 2018 was a very bad year compared to 2017 in many countries. And that would count as emergence or re-emergence of measles. To give you a few more examples that are fairly topical, but I don't want to tread too much on the territory that Colin is going to cover, so I'll go over these quite quickly. Mosquito-borne arboviruses, so that's viruses transmitted by arthropods, or the mosquitoes in this case. We can look at dengue and see how that has changed over time. And you can see the distribution spreading from Southeast Asia and then starting to envelop Central and South America over time. Chikungunya virus, which is another arbovirus, has fairly similar features to dengue, which originated in Africa and then has spread to Asia and then on to South America. And Zika virus, which you'll all remember was very topical a few years ago when there was the outbreak of cases of microcephaly in South America. And Zika virus, which spread from Africa originally, and then into parts of Southeast Asia, and then onto South America as well. So what types of infection emerge? This is another one of the seminal papers in the field, and I think an interesting illustration. And if you look at this to start with, seems quite difficult to make sense of something common about these infections, but there is something common for many of these emerging infectious diseases. And that's that many of them are zoonoses. So what is a zoonosis? Well, as the name probably implies, these are infections which come from animals to humans. And nearly 2/3 of human infectious diseases arise from pathogens that are shared with animals. And most of the emerging infectious diseases identified in people in the last 70 years are zoonoses. So this figure illustrates, I think, a really fundamental principle which will be relevant throughout the rest of the talk. So I'm going to just spend a little while explaining what it shows you, which is the concept of how these zoonotic infections emerge. And it probably won't be very surprising to you, but essentially many of these infections do have a reservoir in wild animals to start with. And they can spill over from those wild animals either directly into humans or more often via domesticated animals, which serve as an amplification of the infectious agent and provide closer contact with humans. Sometimes the spillover is facilitated by vectors. So these are usually little insects or similar creepy crawlies, which can spread the infectious agent from the wild animal quite easily to a domestic animal, perhaps as in the case of mosquitoes, by flying or maybe by jumping or maybe by just simply falling off and waiting for the next host to arrive. And in the case of mosquitoes, obviously they facilitate this transmission happening over longer distances than physical contact. But once these infectious agents have spread into domestic animals, then they can amplify there. So that means more and more animals become infected, often as a consequence of intensive farming and many animals being in very close contact. And then because they are domesticated animals, humans are in contact with them, and the infectious agents can pass into humans. And depending on where those humans are and how close they are together, they may also provide then a source of amplification for further spread. And we'll come back to the different steps here in a little bit more detail as we go on. But if you ever want to read further about these concepts, again, the reference cited here is really a very good one to look at. So if we ask, why do zoonotic infections emerge? We're really talking about the drivers of this, and you don't need to really think too hard to be able to come up with some of these, and I've just illustrated a few of them here. So there are things that we are doing to our planet, and particularly its ecosystems, which are affecting these wild animals and changing their interaction with man and with domestic animals. So if we destroy the habitat of wild animals, then they have to move into different areas. Perhaps they have to move closer to where people are, and that provides more opportunity for transmission. We also do some very strange things with wild animals, and there's an enormous market for wild animals, either for food or for pets, and bringing them directly into contact with people obviously poses a risk of any pathogens that those animals have transmitting. We also have the issue of climate change, and changes in temperature can have really large consequences for changes in the distributions, particularly of vector species, and that can then enable pathogens to be transmitted in places where they previously couldn't be. And then we've been remarkably good at setting up travel networks across the world, and I could be here now in London talking to you, and by this evening I could be several continents away quite easily by catching a flight. And of course that means that any pathogen that I've got with me could also go across that journey, and indeed animals are being transported over these sorts of distances as well. So we've created opportunities to move pathogens around the world very efficiently. So I want to start to get a little bit more specific, and we're going to just focus here on the pre-emergence phase. And I'll give you an example which I think illustrates this nicely. So the pre-emergence is the transfer of the pathogen from the wild animal reservoir into those domestic animals, creating the opportunity for it to start infecting people as well. And in the end of the 1990s in Malaysia, there was an outbreak of Nipah virus, which causes an encephalitis and a respiratory illness. And the situation that created the opportunity for this to happen, and it was mainly pig farmers who were affected, was that there were essentially orchards that were used for cultivating some fruits, and these orchards were also used for pigs to be able to forage and feed, and so the proximity of having the pigs in the orchards seemed like a good idea, but the orchards attracted fruit bats to come and eat the fruit that were in the trees as well, and these fruit bats harbored this virus called Nipah virus. And when the bats' droppings were eaten by the pigs along with the fruit, then that created the opportunity for the virus to enter the pig reservoir, amplify, and then pass on to humans. And actually, this is quite similar to the sort of story that's created in the film Contagion, if you've ever seen that. And moving to something a little bit more sort of scientific around this pathogen sharing between wild animals and domestic animals, people have tried to produce networks of these viruses, and this is an illustration here which just shows lots of connected dots, with dots being different animal species, and showing how many of them link to each other through the pathogens that they share, and particularly very large sharing with domestic animals, which obviously just sort of reinforces this point about the opportunity for pathogens from wild animals to go into the domestic animal reservoir. And it's the drivers of this process of pathogen flow have been very nicely summarized by the UN Environment Programme in a report that was released this year, and I'll come back to it a little bit later. But essentially, you can see from the figure on the left-hand side of the screen this concept that we have the lots and lots of sort of overlapping groups where humans are sharing things with livestock. There's the peri-domestic wildlife, which might include rodents and things which can move around very freely, and the livestock sharing also with wildlife and sometimes directly with humans. And the processes driving pathogen flow here are really all things that we have done to our environment in being a bit careless about how we've used it. So there's an increasing demand for animal protein that results in intensive livestock farming, an unsustainable agricultural intensification, which again creates situations favouring the transfer of pathogens, increased use and exploitation of wildlife. So that's, for example, as pets and as food sources in some countries. And then unsustainable utilization of natural resources by urbanization, change in land use and extractive industries, whether that's deforestation, that's mining, that's building too much. But all of those things having a big impact on the dynamics of these relationships between wildlife and humans and our livestock. And then travel and transportation, as I alluded to, food chains, which are incredibly complicated in terms of where foods are going around the world, that they might start off in one country, they might go to another for processing and then another for consumption. And those countries can sometimes be huge distances apart. And of course, climate change, as I mentioned earlier. And there have been some very nice examples that have really shown the association between, for example, deforestation and changes in pathogen transmission. In Southeast Asia, there is a zoonotic malaria parasite called Plasmodium knowlesi, which is very geographically restricted because its natural host is long-tailed macaques. And these live in forested areas. And as deforestation has occurred, then these macaques have been forced to move closer to where people are living and have increasingly transmitted. And mapping over time cases associated with changes in land use has been done. And actually, although I don't have time to talk about it in detail, some really interesting projects looking at the ecology of this, tracking both the colonies of the macaques, tracking people movements and their interactions and their proximity to the macaques, mapping the land use changes through drones over these forested areas, which really, as a combined source of evidence, show very convincingly that the impact that man's activities has had on really increasing the transmission of this malaria parasite from macaques to humans. And people have actually tried to use some of these features that I've mentioned in predictive models, and you don't need to read all of the points on this, but associating different features with the risk of infectious disease, emerging infection outbreaks. And when those are all combined into a geospatial model, they produce a model looking like this, which illustrates kind of the hotspots of emerging infection. And some of these will not be surprising, and some of them may be a little bit surprising to you. And maybe those are things we can discuss in the questions. But we're starting to get an idea of the types of things that we might be able to use to predict where outbreaks might emerge. So I've talked about this pre-emergence phase, getting into the domestic animal reservoir. And the next thing that happens is the localized emergence. And I alluded to that talking about Nipah virus, but another example would be the sporadic outbreaks of Ebola virus, which also sometimes don't result in very large chains of transmission. But what determines whether a pathogen really spreads to humans is often characteristics of the pathogen, whether it has undergone mutations that allow it to infect humans. Sometimes those mutations may be very minimal. What determines what happens next is often about whether it can sustain human-to-human transmission, and there are chains of transmission. And that's both a characteristic of the pathogen and also dependent to some extent on the population in which it emerges. And one of the big determinants here epidemiologically is the R0. And we've heard an awful lot about this in relation to COVID-19. And one of the things that has spurred is a lot of infographics trying to explain to people what R0 is. So it's the average number of people that one person with a virus or another pathogen infects based in a totally susceptible population. And so you can see this illustrated in different ways for pathogens with different R0 values. I think if you look on the right-hand side here, you see how, for example, the coronavirus causing COVID-19 has an R0 somewhere around 2.5, whereas measles is much higher. Illustrating this in another way, you can see the comparison here. So these numbers are actually really important because the way you need to think about it is that this is the number of people that each infected person can pass or would pass their infection onto. And so this becomes a very, very big difference whether your R0 is 2 or 16 once you go through several generations of infection. So if one person passes it on to 2, then those 2 people pass it on to another 2 each. That's a lot fewer than if one person passes it on to 16 and then those 16 each pass it on to another 16. And to illustrate this, these were some predictions made early in the current COVID-19 pandemic about the number of cases that we might expect based on some different R0 values. And you'll see the range here is very small, from 1.9 to 3.1. But look over the course of just a month or two at the massive difference that you see in the number of cases that may occur based on just these quite small differences in R0. And that's because this is an exponential process. So having a high R0 is probably not enough on its own to turn an infection from a sort of emergent infection into a blockbuster or a pandemic. And that requires a few other things to happen. So from localized emergence, things that are related to the R0, like the ease of spread, whether it's a respiratory spread, whether it's on fomites, which are inanimate objects, so that's often droplets that are stuck onto those, and the ability to form chains of transmission. But also important in causing a pandemic, which is going around the world, is the ability to disseminate that pathogen. And so here I'll talk about some real examples of emerging infections that had big consequences and just illustrate these points a little bit more. So I'm going to start with Ebola and the West African Ebola outbreak, which happened back in 2014. And here you can see the affected countries in West Africa. So Liberia, Sierra Leone, and Guinea. And there were some cases exported as well, but predominantly it was in these areas. And this was really devastating locally in these countries. And Ebola is a hemorrhagic fever. It has its natural reservoir, we think, in fruit bats and spreads from those probably into a variety of other wild mammals. And then from those wild mammals, it may pass into humans through probably bushmeat consumption and also possibly just through contact with things like bat droppings. It's a bit murky exactly how that transmission occurs. And then people get a dreadful illness with this, which you'll be probably quite familiar with the description of, but this hemorrhagic fever. And it's then transmitted person to person by contact with body fluids from those who have been infected. And also it's thought that traditional funeral practices where people would be in close contact with the body of those who died from it may have been a major spreader. And it was interesting that around the time the outbreak was happening, some modeling was being done, looking at modeling the niche for the fruit bat species that were thought to be the natural reservoir of Ebola. And this is sort of done based on the mapping the habitats that these fruit bats would live in and predicting where you might have contact between these fruit bats and human populations. And so independent of the West African epidemic, these models were trained and predicted that West Africa would be a place where you might see outbreaks of Ebola virus, as well as the sort of better known area that was already established before that time in Central Africa. And actually, the West African epidemic was investigated by international authorities on really an unprecedented scale. And I think this investigation was done by CDC, the one I'm showing you here. And they found that they think the source of the or origin of the epidemic could be mapped back to a single village in Guinea called Meliandu. And in this village, they were able to identify the probable first case of Ebola virus of this whole epidemic, which was a child who had actually been playing in a tree, a hollow tree, which was home to bat roosting. And it seemed that that was a very feasible location for the first transmission to have occurred between bat and human in this West African outbreak. And then from there onwards, it was able to spread. And Ebola actually isn't as infectious as we might think. Many people are surprised by this, but its R0 is not particularly high. And it's so when you have an R0 that is relatively close to 1, which means that one person just transmits to one other, then you start to have quite a high element of chance involved in the propagation of virus, because it's not as simple as every person would transmit to one another. It might be that one transmits to 2 or 3 and several transmit to no one else. And so then it becomes like flipping a coin and seeing how many events you get that in a chain. And so you can get chains of transmission that just break through chance, even though you might expect that on average they would continue. And people have simulated this for Ebola, and actually most outbreaks were predicted to be relatively small, although the West African one was sort of one of the outliers in this prediction, with a very large number of cases. And one of the big concerns during this outbreak was about the potential for spread outside of Africa and into major urbanized populations elsewhere, and whether then there would be Ebola, an Ebola pandemic. And people did some modeling of the numbers of flights coming out of the countries that were affected by Ebola, and predicted that there would be internationally transmitted cases if we didn't have restrictions on air travel. I think, to me, the outbreak was very much affecting mainly the poorest populations. This area of West Africa is very poor anyway, but the people who were most affected were those who were least likely to be able to travel internationally anyway. And so I think some of these models were perhaps a little bit unrealistic. And the epidemic came under control once really effective isolation of cases was put into effect, because the R0 didn't have to be pushed down very much in order to stop transmission. Once the R0 falls below 1, then transmission starts to burn out. And so having Ebola treatment centres identifying suspected cases, putting them into those centres and isolating them from the general population was feasible eventually with international help and resulted in the epidemic coming under control. So I'm going to talk next about a different pathogen, and one which will sound a little bit familiar, but this is SARS. So this is not SARS-CoV-2, which is the virus that causes COVID-19. This is the SARS virus, SARS-CoV-1, which caused a global outbreak about 15 years ago. And this is a virus that also has a natural host, probably evolved from bats and then through an intermediate species transmitted to humans. I've put this figure up to just show you that there are quite a lot of coronaviruses. Several of them are ones which are emerging viruses. So we've got SARS-CoV, the SARS virus. We've got the MERS virus, which causes the Middle Eastern Respiratory Syndrome. And we've got SARS-CoV-2, which causes COVID-19. And then there are several other coronaviruses which are circulating in humans anyway, which cause generally cold-like illnesses, although some of them can potentially be a bit more severe than that. And we know the intermediate hosts that have been involved in transmission of the MERS coronavirus, which comes through dromedaries. And we think we know that the SARS virus was transmitted through civet cats in wild animal markets. They cause relatively similar symptoms to COVID-19, although the SARS virus was probably more severe. So this is the global SARS epidemic, and you can see it spread globally with the greatest burden of cases in China. But many countries were affected in terms of cases, but the numbers way lower than we've seen for COVID-19. And this is a timeline of the SARS epidemic. And I don't expect you to read all of this, but I've just, for my reference, highlighted a few key points. So actually, first known case occurred in a region of China in November 2002. And there was quite a while before it was realized that this was a new emerging infection, and that something strange was going on. And then there were, so around the 14th of February, I think it was, that they were, the WHO was notified about this. And then by that point, cases had already spread outside of China. And there was a famous sort of dissemination event that happened in a hotel in Hong Kong, I think as part of a conference trip by a physician from China who had gone there and spread to other guests, who then unfortunately carried it onwards with them to their home destinations. And that resulted in quite a large dissemination. And the other thing that really characterized this, because people didn't know what was going on, was that there was a high rate of transmission to healthcare workers who at that point initially were not using full respiratory precautions to protect them from the virus. But SARS caused a severe disease in the people who acquired it. And so it was relatively easy to identify infected individuals, and these individuals could then be isolated. It didn't take too long from that point to start getting it under control. And you can see really from some of these major dissemination events happening with hospital staff being infected in early March through to the end of May, when really it was coming under control in various countries. And by the end of July, it was really completely ended the epidemic. And that was largely due to being able to isolate individuals who had it because they could be identified, and hospital staff being able to protect themselves from infection from those who were infected. And the SARS virus, so the story kind of ended with the civet cat being implicated, and this was a type of sort of, I guess, raccoon-like mammal that is was traded in some of the live animal markets in parts of China, and the virus was obviously sequenced and examined to try and understand how it had come to be able to spread to humans. And it really had only had to undergo mutations which resulted in 2 amino acid changes to those that were already present in the civet cats that allowed it to be able to spread to people. And it illustrates, I think, a general point with some of the RNA viruses, that they may be more likely to emerge into new infections in humans because they can accumulate mutations a bit more easily than DNA viruses because they often lack a proofreading mechanism which ensures that they try and maintain the same sequence from generation to generation. Okay, I want to move on to something completely different for a minute before we come back to another coronavirus. So many of you will remember that in 2015, we had what was described as a refugee crisis in Europe, with large numbers of displaced people arriving in Europe. And this is a photo that one of my friends and colleagues working in Munich took at Munich Central Station of a lot of a large group of refugees arriving in Munich. And movement of people can sometimes be the driver for emerging infections. And they described an old pathogen returning and being seen with relatively high frequency in their hospitals. Now, I don't know how good your eyesight is and whether you can see anything that strikes you as unusual on this blood film. Probably you're thinking it just looks like maybe a little scratch on the lens that took the picture. But in fact, if we zoom in, you can see these little wiggly forms, which are spirochetes in the blood. And this was louse-borne relapsing fever, or Borrelia recurrentis, which arrived with some of these people who had been living in really quite dreadful conditions as they'd made their way from particularly the Horn of Africa up to Europe. And this was a disease that is very rarely seen nowadays, but was very common. For example, when people were during the World Wars, when soldiers were in very, very cramped conditions for a long time in trenches, and louse infestation was fairly common. And this is transmitted by louse from person to person. So people can be a big driver, but what probably is still the biggest emerging threat is actually probably not even coronavirus at the moment, but something different. And I took this photo, or this photo was taken, I should say, outside the building in which I work in London. And this is the building in which Alexander Fleming discovered penicillin. And the biggest emerging threat for a long time has been hailed as being antimicrobial resistance. And when Fleming was awarded his Nobel Prize for the discovery of penicillin, he said in his Nobel lecture that it's not difficult to make microbes resistant to penicillin, and that people may die as a result of bacteria becoming resistant to it. And I think that was a very important point, and something which perhaps we forgot about as antibiotics were used for all sorts of purposes. But you'll have heard over the last probably 5 years or 10 years, a lot of talk about the threat of emerging antimicrobial resistance. And I've just put a few little illustrations here showing some of the figures that try and make this look particularly scary. I love the CDC report, which describes the threat levels of different resistant pathogens, really like top trump cards. And you can see the reports of data from the UK with increasing levels of notification of antimicrobial resistant organisms, particularly those resistant to carbapenems, which is really, those are our biggest worry in terms of the organisms becoming almost untreatable. And these trends have been apparent across Europe, that we have antimicrobial resistance that has spread dramatically. This illustrates third generation cephalosporin resistance in E. coli, and ECDC, the European Centre for Disease Control, has some great illustrations of this. So this has been going on for some time. It's been very much overshadowed by the current pandemic, but it's still going on. The pandemic may even have fueled a greater increase in antimicrobial resistance. I think only time will tell. And this is driven only by our use of antimicrobials in our patients when we do this unnecessarily, but it's also driven very substantially, probably mostly, by antimicrobial use in agriculture for these animals that we are intensively farming in order to get the greatest yield of meat from them. We have given them antibiotics, and those antibiotics pass into humans as well. So let's come back to COVID-19, and this is the Johns Hopkins dashboard, which shows the global cases, global deaths, and all the other sort of tragic figures that are associated with this pandemic. And this snapshot is from last night. It's probably already gone quite a bit higher by this morning. It's astonishing to think 65 million plus cases, more than 1.5 million deaths attributed to this virus already. So there's quite a lot to think about here, which also illustrates the points of why infections emerge in the first place. This is just a brief timeline, which only runs up to October, so already out of date, but I think summarizes some of the early events well. So at this point, it's thought that the earliest case occurred probably back on the 8th of December, and that then it was realized there were clustering of cases of a strange pneumonia that was severe with unknown cause right at the very end of December of 2019. And in January, it was announced that this was due to a novel coronavirus. And a lot of things happened in quick succession in January. Unfortunately, cases started to be exported from China. It was confirmed that there was human-to-human transmission occurring. And Wuhan City, as you will know, was locked down as a result, because that was the center of the epidemic occurring at the time. And by the end of January, WHO had declared a public health emergency of international concern. The risk level was raised over the course of February, and in February it was also decided by the international committees that the disease would be named COVID-19 and the virus was going to be called SARS coronavirus 2 or SARS-CoV-2. And in March, WHO defined the pandemic, and from there on, well, you've all lived through it, so you know what's happened. And I showed you the most recent number of cases on the Johns Hopkins dashboard. We still don't know the origin of this virus exactly, but you can see this kind of family tree of viruses showing the relationships between the current SARS-CoV-2 lineage, which are indicated around the middle of the figure here, and that it has greatest similarity to some bat coronaviruses and to coronaviruses that are present in pangolins, which are quite a cute little sort of scaly mammal. But although there's been a lot of speculation about which one is the origin, I think it's safe to say that at the moment that debate is still not fully resolved. And you can see that it has relationship to the SARS coronavirus that caused the SARS outbreak back in 2003, 2004. But it's more closely related to these bat and pangolin SARS-CoV-2 lineage viruses. And I would actually recommend, if you're interested, have a look at this paper that is referenced here, that I'm going to talk a little bit about their thoughts on how this came to emerge. because actually it's quite a light read. It's not very in-depth science in it, but it explains some of the factors that contributed to this emergence of SARS-CoV-2 and the COVID-19 pandemic. So these all recapitulate things that I have mentioned already that create the conditions for pre-emergence and emergence into the population. So we have bats and pangolins with the most closely related viruses, which have been in contact with each other and in contact with humans in a variety of different scenarios in the part of China where the virus emerged. So the pangolins have certainly been in wildlife markets and have been used for meats and have been used in traditional medicines. And the bats also have been in contact probably with humans around sort of farming activities and agriculture, and perhaps provided opportunities for transmission through other as yet unidentified mammalian reservoirs. And so this creates the opportunity for the virus to jump from one species to another and to get into man. And it seems that this jump happened in an area that was quite densely populated, and also whenever it happened, and we don't know quite exactly when the very first jump happened, we only know the first identified case, but around the time of that case occurring, there were quite a lot of human activities which provided a great opportunity for the virus to amplify within the human population. And this was the gathering together of quite large numbers of people, not only because these were urban centers, but also because of a number of cultural events that were happening around the time, which resulted in festivals and large numbers of people congregating and socializing more than they would normally do in an average week. And so the early days of the pandemic in China are depicted here, and you can see how cases started to go up after this stage. You will always expect a lag in cases identified behind transmission, but you see quite rapidly they're increasing, and the number of deaths starts to increase as well. Although this largely shows you just in a different format what I was saying from the previous timeline figures. And then the authors of this article suggest that the epidemic phase was very much facilitated by not just local events, but national events in China, with people coming and going from this province as a result of one of the big national festivals. And also, along with that, a huge number of students who travel in and out of the province in Wuhan in order for education. And I think there were not only events that brought a lot of people together through some of the festivals, but then there were events that disseminated a lot of people elsewhere within China very rapidly. So those two events happened sort of one after the other, creating a perfect storm in a way for people to contract the new infectious agent and then to spread it outside of the area where it had locally emerged. And you can see by the end of February, the numbers of cases that were occurring around China. So essentially they'd spread to every district of China, although still focused around Wuhan as the largest number of cases. And then the final phase that turned it into a blockbuster was the pandemic spread, and that's really international dissemination. And unfortunately, there was a lot of travel to and from China during this period as well, internationally. And you can see here by the end of February, the number of international dissemination of cases that were identified. But of course, what we know now is that this probably is a big underestimate of the number of transmissions, because the range of symptoms from this virus can be from asymptomatic through to severe disease. And case definitions at the time, looking for people with quite severe illness, probably would not have picked up those who were less symptomatically infected. And so that provided the opportunity then for an enormous amplification of transmission around the world. And so then we end up where we are now. As you know, that this pandemic has reached pretty much everywhere and caused a vast number of cases. So I want to finish by just considering steps for preventing the next pandemic. And you might think that we should be on the lookout for this, but how we do that is actually a little bit challenging. Here you have a picture of one of the George Bushes through his binoculars, but actually the lens caps are on, so he's not seeing anything. And some of our efforts to predict the next pandemic are a little bit like this. I see that Colin has appeared, so I'm going to speed up a little bit. So surveillance has dramatically changed over time, and we now have amazing molecular surveillance available to us. So we can do a lot, but how we use it is still challenging. For a long time, there had been, well, over the last couple of decades, there had been a program trying to predict emerging pandemic threats. But this program, ironically, was shut down. Its funding was ended just really weeks before the onset of the SARS-CoV-2 pandemic. And PREDICT2 was the last strand of funding from USAID for this emerging pandemic threats program. PREDICT, the whole program running from 2009 to 2019, spanned 60 countries and was focused on building capacity as well as undertaking surveillance for potential zoonotic pathogens. And they identified a large number of novel virus species, but it was impossible to say how many of these really had the potential to become zoonoses. In fact, only one conclusive zoonosis was identified from this enormous effort of screening. And of course, the only way to tell which of the very many zoonotic, potentially zoonotic mammalian viruses will transmit to humans is to observe for a human infection at the moment. So the UN has released this report from the UN Environment Programme, Preventing the Next Pandemic. It emphasizes the importance of early detection in being able to reduce cases. That is taken as read, really, that the peak of cases will diminish if we can detect early and we can put control operations in place early. But to me, reading this report, the big message, which they don't state very boldly, but once you read it, you realize that actually we brought this on ourselves, not in a biblical sense, but in a sense that we created the conditions that allowed this pandemic to occur. And this is through those anthropogenic drivers of emerging infection. And if we want to prevent the next pandemic, we need to reduce demand for animal protein. We need to introduce sustainable agriculture practices. We need to decrease the use and exploitation of wildlife. We need sustainable utilization of natural resources, rethinking how we use land and how we extract what we need from the land. We need to rethink the need for us to travel so widely. We need to rethink food supply chains and probably make food much more local and sustainable. And we need to tackle climate change. And these are all big issues, because if we don't deal with these, we're going to have more and more pandemics of emerging infectious diseases. So I'm on my summary slide. I'm sorry I've gone a bit longer than I had planned, but there are many emerging infectious diseases. Most of them are zoonotic. They often occur because of complex changes in our environment. Sometimes these are very complex, but mostly because of what we've done to the environment. There's now a lot of global interest in identifying and predicting threats to prevent the economic consequences. But if we really want to prevent future pandemics, we need to change the way that we, humankind, interact with the environment and with animals. And I'll end there. Thank you.
Thank you very much, Aubrey. Might I just ask an enormous question quickly? Why do you think many pandemics and epidemics resolve? Where is it they go to? I know it's a massive question and it's a big generalization, but you've explained how all these things arrive. Do we know roughly what it is that makes them disappear, some more quickly than others?
Okay, well, that's a big question. I don't think there's probably a single answer to that, but there are some very good examples. So immunity is one, herd immunity, because once enough people in a population are immune, that means the transmission, the R naught, will fall. Well, R naught applies to a completely susceptible population, but the R number will fall below 1 when enough people are immune. So a good example of that is Zika virus. We heard, I don't know if you're going to talk about this, Colin, but we heard a lot about the microcephaly kind of epidemic happening in Brazil. And then there don't seem to be so many cases happening anymore. And it seems that in a way, a lot of people became immune to Zika virus after being infected, and that was enough to reduce transmission to the point that it's almost sort of died out. For others, obviously, we put control measures in place. So those control measures might be trying to kill mosquitoes if it's an arbovirus that are transmitted by mosquitoes. It might be isolating infected individuals and breaking those chains of transmission in that way. So not one size fits all, but yeah, there are many reasons for it.
Thank you very much. I can't find anything else on the chat at this point. So I think perhaps we should move on and I can do my presentation. So thank you very much for yours, which has launched me into talking a little bit about something that is related. So let me see if you could withdraw your screen, Aubrey. I'll share mine. Thank you. So I'd like to give a presentation which is sort of posing the question. I'd like to talk to you about our planet's biggest killer, but is it? And if not, why not? Because it fits in very well with the presentation we've just heard, where Aubrey Cunnington explained that the anthropogenic drivers are predominant in pushing emergent diseases forward. There is a little beast in the middle, or a set of little beasts, and I'd like to talk to you about these. I'd like to talk to you about mosquitoes. My name's COLIN MICHIE. I'm currently helping run a school of medicine in the University of Central Lancashire, and up in the Lancashire Dales, one does find some of these, but not terribly many of these. This is a mosquito, and the group of mosquito insects is really quite important because they are extremely dangerous. And I'd like to spend some time explaining why that is. Essentially, mosquitoes are always likely to pose a major threat to health. They are weaponized for change. They are extremely capable, and they have wide capacities, ranges, and the diseases they carry by various means are expanding as climatic change evolves. So the problem with the mosquitoes too is that they're becoming resistant to insecticides, and that problem makes them very difficult for us to manage. Historically, controls of mosquitoes included the way people designed their houses, their vernacular architecture, the use of bed nets too. And I'd like to point out at the end of my lecture that in fact, there are reasons for thinking that maybe we don't pay quite enough attention to those old adages and those old ways of doing things, because certainly insecticides aren't working quite so well. And so I'd like to start with a bit of history really. And this object, this is the head of a glass walking cane, which was passed down to a member of my family who lived in Essex in a little village called Castle Hedingham. And this is a long piece of glass. It was probably made in the late 18th century, and it's called a witch's walking cane. And the reason one had a witch's walking cane was to protect you from the ague. the ague was a frightful infectious condition. It came upon you suddenly, and it caused very severe fevers. Sometimes it caused death. And so people in 18th century England had a number of ways of trying to protect themselves against the ague. The ague was particularly common along the eastern parts of England, and in fact up on the northwest too, because it was in fact malaria. It only got identified as malaria in the early 19th century, but essentially for many years, items such as witches' walking canes were used by families to try to protect themselves. And what one had to do was put the witch's walking stick between the fireplace and the bed, and that way one would not catch the ague at night. The big mortality picture, standing back and looking at mosquitoes, is that there's many trillions of mosquitoes across the globe as we sit here listening. They are unrelentingly out for a blood feed. So the females are the ones that do the blood feeding, but over 50 mosquito species carry pathogens. The females carry pathogens. It includes 5 types of Plasmodium, one of which we've heard about already this morning. But essentially, the females are unrelenting hunters. They are out for their 3 to 5 milligrams of blood, and with that, they will lay eggs and pass on their species. But our human global defense budget, if you like, against mosquitoes is relatively small. It's only estimated at £9 billion globally, so that although we have a lot of these animals out there, and although there are many species and many types and a large number of pathogens, our defense budget is relatively small. Fortunately, the one big index disease in which we're all interested in as pediatricians is malaria, and this being an affliction very much of children in southern parts of Africa, the sub-Saharan African areas, and thankfully the deaths are falling, but it is still very high, and it's still estimated at being over 400,000 deaths a year. So essentially, the index disease about which we're all worried, the killer disease, which is largely a type of Plasmodium called falciparum, is becoming less common for a bunch of reasons. But as the animals that transmit these pathogens, mosquitoes remain powerful, and indeed they have been throughout human history. And Charles Darwin once said this, and this is relevant when you talk about all of the intermediaries in Aubrey Cunningham's drawings and diagrams and in the story here of mosquitoes. Charles Darwin said, "It's not the strongest of the species that survives, nor the most intelligent that survives. It is the one that is most adaptable to change." And so I've put on this a number of illustrations of how malaria has compromised major military efforts through the ages. So malaria down on the bottom left here knocks you flat. Keep covered, use your repellent, because for troops going anywhere into tropical areas, malaria was a big threat and still is. The one below here in relation to your organization relates to a Second World War cartoon, as does the part up in the top corner, and you'll recognize the style of this, but this was Dr. Seuss's first job, because Dr. Seuss, with whom we are all well acquainted as pediatricians, actually drew a set of adverts to try and make troops in Vietnam aware of the risks of mosquitoes and malaria. So the problem really is mosquitoes are incredibly adaptable and can be quite threatening as a consequence of that in ways perhaps that we don't understand. And the first point to make is that mosquitoes were here before the last major extinction. So mosquitoes fed from dinosaurs. And we know that mosquitoes fed from T-Rex, for instance, and that indeed the dinosaurs were afflicted with plasmodia spread by mosquitoes. So when you see the mosquito, as up on the top right here, embedded in amber, what one is seeing is a fossil, not only of the mosquito, but of a disease-spreading organism, a pathogen-spreading organism that was doing the job very well. And heaven knows how they managed to bite through a T-Rex skin, but they did, and the dinosaurs suffered. And although the pathologies were relatively small, we don't think that they were lethal diseases for large dinosaurs. We don't actually know, but through the major extinction, mosquitoes persisted. So they are pre-mammalian, and therefore very adaptable and are quite agile in the way they can adopt different hosts. So do we salute it, or do we just splat it? mosquitoes have a set of common features. They home in on heat, carbon dioxide, lactic acid that we are all passing out into the atmosphere around about us. And when you ask who is more likely to be bitten by a mosquito, there is one or two clues, but in fact, blood group O, as sung by the Disney tune for The Bug's Life, is in fact slightly, it does have an element of truth. So those with blood group O are slightly more predisposed to getting bites and get more bites than others when exposed to mosquitoes. And what mosquitoes do is behaviorally very interesting, but the males who do not bite us and do not bite and blood feed tend to cloud around the potential victims. So there are photographs in some areas of clouds of several hundred mosquitoes, male mosquitoes, over a person waiting for a female to come in and bite that person. So essentially the males will hang over you, they will buzz around you, but what they're actually waiting for is a female mosquito. Having mated, the mosquito will then bite and get her first blood feed. And one set of semen is enough for the female to make several sets of eggs. What she is restricted by is the blood feed. So she will make a batch of eggs straight after each feed, 25 to 50 eggs. And those eggs tend to hatch out in a week to 2 weeks. And one can then have another batch of mosquitoes en route very rapidly and very efficiently. So the problem of how agile they are has been solved really by a number of behavioral mechanisms that mosquitoes adopt. And the reason they are so dangerous is largely because of the payloads. Now, this focus here is very much on the pathogens. So that's one aspect, but of course, for most of us, pathogens aren't really top of our mind when we get bitten by a mosquito. Mosquitoes cause significant pathology just through the bite. So there's irritation, inflammation, sometimes a degree of allergic reaction around a bite. And of course, if your skin has a bacterial species or two on its surface, it can get injected by mosquitoes. So there are cases, albeit rare, for instance, of people contracting tetanus from a bite from a mosquito. But essentially, the majority of pathogens typically infect the salivary glands. They're injected along with anticoagulant as the female feeds. And the pathogens aren't inert. They can influence the way the mosquito feeds. So for instance, plasmodia are known once they are within a set of salivary glands in a mosquito to make that mosquito feed more often, for instance. And the rate of feeding will go up as the temperatures rise too. So there are issues with how the various pathogens interact with the host mosquito. And it's also been hypothesized recently that microfilariae, which are carried within mosquitoes, not so much within the salivary glands, but within their body cavities, are capable of transmitting genetic material between mosquito species, which makes both the microfilariae and the mosquitoes a lot more adaptable to a variety of environmental situations. So what pathogens are commonly known about? Well, human pathogens carried by mosquitoes include the plasmodium malaria, yellow fever, dengue fever, Zika, as we've heard, West Nile virus, as we've heard, chikungunya, a set of encephalitides, microfilariae. Recently, rickettsia have been found being transmitted by mosquitoes too, tularemia, Rift Valley fever, and in fact, there's a fair number of others particularly from Australia. There's a set of slightly unusual organisms transmitted by mosquitoes there, but they are all going to fall within this sort of pattern of disease manifestation. But it's important to remember, as Aubrey pointed out, domestic pets, sheep, horses, birds are part of transmission, and indeed they are infected and bitten by mosquitoes in much the same way. And mosquitoes sometimes can share plasmodia. And it's known, for instance, that if you are in a jungle area in Africa, you can contract a plasmodial species from a large monkey nearby, because the parasitic mosquito may have in fact bitten a monkey just before it bit you. So sometimes you will be transmitted an organism which is not likely to cause illness, but can be detected. And the PCR systems that are now used to analyze pathogens carried by mosquitoes, carried out in the Caribbean, have identified some individual mosquitoes as carrying several viruses. So it isn't one virus per mozzie. In fact, they are capable of more than that. So these mozzies have a major skill set. And what impact have they had in terms of our past? And I think the best way of illustrating this is to look at the effect of falciparum malaria, because it kills human beings when they're young and before they've had children. And as a consequence, Plasmodium falciparum, as one of these pathogens that mozzies transmit, is capable of changing our population genomes. And it has had a massive effect on our human genome that we know about. I mean, this is true of other infectious agents too, but in terms of this one, we know more about what effect it's had because it influences red cells and endothelia. So hemoglobinopathies, sickle cell and thalassemia fall into that, but think about all the variant hemoglobins. They too are all adaptations to the fact that falciparum killed our predecessors. Glycophorins, PKD and G6PD, these are enzymatic problems that develop within red cells. They are related again to malaria, and the Duffy blood group and other blood group systems have been distorted by this one pathogen. So mosquitoes have, within their skill set, managed to alter our genome significantly. And although we will all be told at medical school that mosquitoes can fly 400 meters and then stop and that's it. And so they always expand and contract within their 400 meters as a single individual. This is in fact found not to be true. And recent studies in Africa have shown that if a lot of mosquitoes after a feed will fly straight up. The reason this was noticed initially was that everyone knows that a number of birds fly quite high and seem to get quite good insect feeds. And when you look at what the birds are eating, it's largely mosquitoes. And mosquitoes fly up and then catch winds. And on a wind, they can travel several hundred kilometers. So for instance, there have been outbreaks in the Middle East, which have been attributed to mosquitoes that have blown across many hundreds of kilometers from Africa. And in the south of England, in terms of an infection called blue tongue in sheep, it's known that those mosquitoes have blown across the channel with great ease. So essentially, it is known that the mosquitoes will move. They don't have to be carried in water as used to be taught, but in fact, they are capable as adults of flying. Their genomes are quite large, 270 megabytes thereabouts. They've got a large number of genes, about 13,000, and there is interspecies exchange of genetic material. So these creatures have got a large and extremely flexible genome and can withstand an awful lot of change. As an example, there's a rather charming piece of entomology that relates to the Metro and the Second World War. But during the bombing, a lot of people took refuge in the undergrounds in London, and they were followed closely by a culicine mosquito that used to bite them up on the surface. But the culicine mosquitoes went into the undergrounds too, and they've actually stayed down there. And they have bred down there since, and they now live on the other mammals that inhabit, as well as humans that use the underground. And so if you look at the mosquito subspecies that is there now, that is slightly different to the mosquito that was its predecessor sort of 80, 90 years ago. Essentially, the culicines have changed. There is a subspecies. And one of the entomologists at the London Zoo has always joked that maybe at some point we'll get a subspecies for each set of Metro underlying tracks. So essentially, the mozzies are capable of adjusting and actually becoming subspecies within a very short pace of time. But the expansionist capacities, of which the Metro is one example, is also shown by what we were described earlier today. The climatic change and deforestation is going to increase the range of pathogen-carrying mosquitoes. They are going to want to find new victims. They need their blood feeds and they will expand. And if we've taken their natural habitats away and the natural ecosystems in which they inhabit, the numbers will surely decrease, but we will have species that are going to spread. It is known, for instance, that natural disasters increase the rates of all arbovirus infections. So yellow fever and dengue in particular are much more marked after a natural disaster in a tropical area. Malaria and filariasis likewise. So that when you stand back and look at it from the sort of population point of view, highest disease rates from mosquito-spread pathogens are likely to be found in poor urban populations who are exposed to trauma or to business or population movements. So if you look at the multiple pathogen carriage issue, which is really frightening, and here's the continent of Africa, and we know from the top map that there's an overlap of a set of diseases carried by mosquitoes. But when you look at individual reports, as in the lower map, you will find that they coincide. So chikungunya, dengue, yellow fever, and Zika can be identified within the same villages, within the same groups of people, spread by different mosquitoes, sometimes the same mosquito, as has been proven as with the Caribbean studies. So essentially, there's multiple pathogens being spread by this one set of vectors, the mosquito. And if you look at arbovirus geographies in other ways, again, you can pick up on some fascinating pictures, which we are sometimes not terribly aware of in Europe. So the top right map shows the global areas likely to be affected by dengue fever. Dengue fever, again, is something that afflicts a lot of children, and it's been discussed in the EIP meetings before, and the experts are largely from Sri Lanka, because these folk down here get very exposed to dengue and have had it for many years, and they've become expert at treating children with dengue. But essentially, dengue zones are likely to increase, and these wavy lines indicate that really things may change. So it depends on isotherms, and if your isotherms move, as they will with climatic alterations, it is likely that the host mosquitoes will move and this disease will become more prevalent. And there are predictions that dengue will be much more common in Europe in future. Likewise, if we look down here at the set of continents here, we're looking at cases of Zika, and Zika has been spread, and I'll show you another map of Zika in a moment, but essentially Zika case numbers were originally really quite low, but became very rapidly increased as the mosquitoes that transmitted it moved. And so here we have another way of depicting that. So Zika was originally described in a forest in Uganda, which is where these two red arrows originate in Africa. It spread across Africa to the west, but it also spread into the Pacific Rim. And when it came into Brazil and into the Caribbean basin and up into the North American continent too, a number of patterns in connection with microcephaly and phytopathies were described. During the course of this migration, the issue was partly the fact that mosquitoes were moving and were transmitted either in aircraft or by local conditions within these points, but also because the Zika virus was changing. So there was a genetic change in the virus by the time it got to the Pacific Rim, further changes by the time it got over here, so that the pattern, as Aubrey described earlier today, was different and essentially appears to have resolved as a consequence of herd immunity in this area, certainly around here, but Zika can still be detected and was still being found in the Caribbean earlier this year, so that there are cases. It could spread again if it changed into a slightly different organism to which we had no immunity. Dengue potentials are shown in two maps here. So the map A at the top is the estimated population at risk in 1990. The bottom global pattern is for 2085. And so one can see that essentially the amount of the number of folks and the amount of land that is going to be covered by mosquitoes that can spread this organism are going to increase dramatically. And we probably all need to learn a lot more about how to manage dengue. What's worrying too about mosquitoes in reflecting on their ability as vectors is that they become resistant to insecticides very rapidly. Most insecticides are neurotoxins, so they will act on something to do with the nervous system in the mosquito. A large number act on a voltage-gated sodium channel, such as DDT and the pyrethrins, and acetylcholine receptors and GABA receptors in mosquitoes have become targets for new and insecticides too. But the problem with the neurotoxins is that they can have quite a wide-ranging vector base. So in fact, DDT, which was a terrible insecticide from the perspective of the environment, but for the mosquito mosquito control, it was fantastic, because for 50 years it controlled mosquitoes pretty effectively and prevented a lot of mosquito spread disease. but essentially DDT is now used in very restricted areas because of its side effects, both on us and other insect species. But we replaced this with pyrethrins, and pyrethrins have become a problem because of resistance they developed within a number of mosquito species and mosquito groups. Acetylcholine receptors, these are new insecticides. Again, insecticide resistance has been detected. There's further resistance, not only on behalf of the nervous system of mosquitoes, but metabolic ones. So like ourselves, mosquitoes have cytochrome P450s and other sets of enzymes that are capable of damaging and reducing the effect of anything that they are sprayed with. And so cytochrome P450 needs to be influenced as well. So there's a new compound which has been tested and tried. It now has had some 12, 15 years experience in the field. PBO, piperonal butoxide, reduces cytochrome P450 activity in a mosquito. And so if one uses pyrethrins plus PBO on a net, for instance, or as a spray within a hut, within a homestead, you can actually prevent the metabolism of the insecticide, and the insecticide becomes much more potent. So this system is now being used to modify our use of insecticides in mosquitoes, but the resistance to them is rapidly developed. And the reason we know quite so much about this is largely due to genomics. At one point in time, we would compare mosquito survival rates or drop rates with and the effect of a spray of an insecticide, for instance. However, work from the groups like in the Sange laboratories in Cambridge has completely turned this upside down, because by collecting mosquitoes and doing genomic signatures, one can analyze what's going on in that mosquito with respect to the pathogens it carries, the insecticide resistance genes it's expressing, and indeed its predecessors and its links to other populations in so much greater detail. This requires high-tech laboratories and high-tech computing, but genomic signatures are now becoming a way to study mosquitoes, which is very interesting. Many parts of tropical medicine are having to adapt extremely rapidly to cutting-edge genomics. So just as an example, on the map of Africa down here, there are amplifications and resistance genes demonstrated in different colors within these circles. And what it's possible to see demonstrate with a broad paintbrush is that there are different colors here. And essentially the colors within this sort of far left hand around Gambia and Liberia here are quite different to those that are overseen in the south of Sudan, or perhaps in the DRC, and Gabon and the sort of Central African areas here. So essentially, different groups of mosquitoes are exhibiting different patterns of resistance to insecticides, and these patterns spread. They share these abilities to avoid what we are using to try to treat them. So how do we tackle mosquitoes? Well, clearly you want to avoid the bites, and to use a famous phrase, draining the swamps is the clear way to get this done. Now, draining swamps is quite well established, and the Italians did it. The Italians named malaria malaria way back because of problems that beset those who lived in Rome. The swamps around Rome were full of mosquitoes, and a physician called Celli, who was very adept at figuring this out, thought that draining the swamps was the first pattern. In those days, they believed that malaria, the illness, was probably caused by a miasma. The link to the mosquito wasn't established until 1850s, and that link was made by this Frenchman called Beaupetit. But essentially, prior to that, Celli and his colleagues had figured out there was something about marshes and the marsh miasma that was bad for health. And so draining swamps, which effectively withdrew mosquitoes, was an early treatment and is still a practiced one. Beaupetit was a Frenchman. He was practicing in the north of Venezuela, and using his microscope, he figured out that mosquitoes had something to do with yellow fever. And he actually wrote, and sadly he published it in the local gazette rather than an international journal, but Beaupetit observed that the local stripy-legged mosquitoes were the ones that caused yellow fever. It took another 50 years for the rest of the world to agree to all of this and sort of formalize that relationship. But Bopetit, meanwhile, together with his colleagues, drained swamps in Venezuela because he thought this was the best way to get rid of the mosquito and stop them biting. So the problem about draining swamps is largely one of public health and engineering. On our cells, there's a bunch of surface protections we can use to deter mosquitoes. To be frank, they aren't brilliant and they don't work for long. And a lot of them have side effects on our skins. So essentially surface protections aren't terribly well developed yet. And this is an area which does require a lot more innovation, shall we say, because of the current problems we have with the agents available. What's particularly interesting is the business of vernacular architectures. So essentially, humans have lived close to mosquitoes since humans began. Since humans first existed in communities, there have been mosquitoes there. And so humans have developed ways of building their homes and perhaps planting things around their homes to protect themselves. This is an area about which we know remarkably little. And so I'd like to talk about that in a slide a little bit further on. We can use nets, insecticide, and nets have been in use since way back 3,000 years. Nets have been used and to keep mosquitoes away from beds. However, they've never been favored and they've never become terribly trendy, except in some parts of Southeast Asia. And certainly, again, I'll explain and show a demonstration of that in a moment. Insecticides, I've discussed very briefly, and there are natural agents such as Bacillus thuringiensis, which you can put into the mosquito's environment. This organism can be sprayed into ponds, into small amounts of water. It can be used to coat a homestead wall, and it infects the mosquitoes to the point that they don't multiply very effectively and they certainly can't lay eggs. So infecting them with something to which they don't have a very good defense does seem to be a useful short-term strategy. In terms of its wider side effects, this hasn't been explored and it hasn't been used on very large scales in Africa or the Caribbean as yet. The other major systems which are now becoming more and more popular and are already commercially available is to develop a system whereby one can release infected mosquitoes or sterilized mosquitoes or mosquitoes with a lethal mutation into the population of wild mosquitoes to then adjust that population and stop it multiplying. So again, I'll discuss that briefly. This is the range of our armamentarium to which we are dedicated to developing really and have to improve if we're going to stop the advance of mosquito spread pathogens. So home and bed nets. The vernacular architecture systems. These systems, living in a mud hut is actually not quite as uncomfortable as it might seem, because a traditional African mud hut is remarkably mosquito free. And the tricks are to make sure that there's no water around the hut, that the eaves are sealed. So mosquitoes will almost always fly in and fly up under the eaves. And it's very important to seal up around the eaves and essentially trap the mosquitoes there. Doors must be low and shut at nighttime, and there shouldn't be windows because that way the mosquitoes won't get in. And if you have a nice polished floor, and cow dung is traditionally used for that, but mud from different places will work just as well as the occasional fire inside is very good at keeping these little huts mosquito free. So this is how insects are being kept away from humans from our perspective for thousands of generations. If you go to Javanese architecture as here, or Japanese architecture similarly, again, the principle of having a high ceiling, very few windows, everything shielded off and sealed under the eaves is crucial. And again, this is very protective. In terms of local environmental changes, people in India and Sri Lanka use small fish. So wherever there is an irrigated paddy field or wherever there's a collection of water, they will put in fish to try to eat the larvae of mosquitoes. This is fairly effective, and it's sort of a natural system which has been developed to some extent by some group communities to great extent. And of course, there's some vegetation which mosquitoes don't like. And again, there's a great folklore about this. The value of nets, though, as shown here, has been known for hundreds of years. And the Japanese net, which we know quite a lot about because it's been depicted in their pictures and etchings for several hundred years now, has been well developed. So since the Edo dynasty, this business of protecting both children and adults is well developed there. And nets are so much more effective if you can kill the mosquito when it sits on the net. So the mosquito will fly in, if you're unlucky, will sit on the net and then die because it's become poisoned by an insecticide. Excuse me. So pyrethroids plus PBO have become a way of protecting nets very effectively. The lethal mutation system allows itself to spread by spreading something into a population of mosquitoes which prevents them breeding further. So one takes a token female, and that's the system that's been used so far, bearing a lethal mutation, and multiplies her up in an artificial situation and releases large numbers of herself and her cloned progenies or sister progenies into a population where they breed with the males or try to breed with the males or distract the males, but essentially reduce the breeding population's ability to expand. Lethal mutations are in their infancy, and because of the ethics of releasing lethal mutations into environments, they've been used least frequently. What's been used a lot more often is either releasing sterile males or infected males, and they work on the same principle here, whereby sterilizing a male and releasing large numbers of them will cause changes in the population, because essentially a sterile male and a wild female will not have any viable offspring. However, this system takes a little while to work, and one requires large numbers of sterile males. However, one can buy them in certain places in the world already, and this system has been tried and tested and is very effective in small areas, and is particularly useful on islands, for instance. And the Australians have used a similar system to this, whereby rather than using a sterile male, one uses a male that's been infected with an organism called Wolbachia. Wolbachia does much the same job as sterilizing the male. It makes it impossible for that male to have infants and to pass on any effective semen. So essentially, an infected male with Wolbachia will have the same effect on a large population. And that particular system is now probably much more common throughout the Pacific, and it is spreading into the Caribbean too. So there is an issue here which comes home to us now as pediatricians. Most people don't worry about mosquitoes, and we need to get this message about how dangerous they are, preventing and protecting individuals, because essentially the mosquitoes are very good at getting around everything that we manage to throw at them. What are the risks of our various controls? What are the risks of insecticides? The risks of DDT are pretty well described, and thanks to Rachel Carson and colleagues since, essentially those particular insecticides are used relatively infrequently, although they still are used. What are the risks from burning coils inside houses? Again, there's a set of individuals who think this is an incredibly dangerous thing for us to do, but they are still widely sold. What are the risks of engineered mosquitoes? So balancing up our various control methods is going to be an important one, and the population scrutiny of these various methods is kind of in its infancy. And we need to bear in mind, clearly now that we know more about this problem, the non-human risks. What about the other insects? Bees are compromised by things we have done from our agriculture, largely related to insecticide use, I suspect, but possibly other things too. But essentially, we have to be very careful if we're going to try and control one lot of insects that we don't kill others. Beetles and other insects, the One Health approach, are clearly very important, and we don't know very much about this problem. We only know something when we lose it, and right now this is an important problem with bees in particular. And we always need to bear in mind the harms and benefits to our domesticated animal population. What will happen to the sheep, the horses, and small animals? So, for instance, in the Caribbean, sheep cannot be kept there because of a number of mosquito-borne illnesses which are lethal. A lot of the horses die there because of West Nile virus, and West Nile virus is incredibly common. And a lot of small animals, most people's pets, have to have an incredibly expensive procedure of vaccination and treatment undergone regularly because of lungworm and heartworm, which are essentially microfilariae spread by mosquitoes. So essentially, the risks to the animals around us are high and sometimes not awfully well described. So just to go back to the communication, I think Dr. Zeus really had it, and his little pamphlet, which consists of about 3 pages, and it's well worth looking, is all about Anne Anopheles. This is Anne. She drinks your blood. She's dying to meet you. surveillance is another part of this component. I mentioned earlier, genetic surveillance is actually crucial. We're going to need lots of genetic surveillance in the future in order to tell the story of what is going on very accurately on the ground. And this will allow us to manage insecticide resistance and evolution in the mosquitoes too. What species are where? What are they carrying? The answers will be here, but this is an expensive process and it requires a lot of good collaboration. In the meanwhile, those of us that aren't sitting with a Sanger network close at hand can help. And there's a series of citizen science projects, and these are great for all of us to consider in terms of asking local groups, local communities, local schools to get involved with. But Network is one of the older ones, and if you go to the website for Network, they will explain how to move on from there, and they also link together a whole series of these citizen science projects. But there's now a mosquito alert app which has come from European colleagues, and that is here on this website. So essentially there are sets of surveillance systems which are available and are quite easy for any of us to use, which will be useful. They marry together in the sort of same manner as the infamous touch and trace system, the ability to say, this is this mosquito, I saw it here on this date, and can it be identified? So in conclusion, mosquitoes are agile predators of our species. The pathogens they transmit are genetically adaptable too, and older methods of avoiding bites from mosquitoes, architecture and nets, for instance, certainly need revisiting in conjunction with modern methods. And we need to develop our genetic methodologies in order to outpace the mosquito and protect our vulnerable populations in future. I'd just like to thank the organizers for inviting me to this meeting. I'd like to thank my university who supported me, and the colleagues and students who challenge and deliver great questions. And with tongue in cheek, I show you the what was to have been the Mississippi flag. An individual from Louisiana wished the mosquito to be on the new flag for his state. This was banned within a very short space of time, but I rather love it because it illustrates that certain parts of the world do regard this as a major threat. So thank you very much for your time, and I'd welcome questions. So, Aubrey, we have one question here. How do you think climate change will impact emerging infectious diseases in Europe? Wow, that's a big one. You take that one.
Well, I think we could probably both speak to that one, Colin, and you've alluded to it a little bit in your talk already. And I guess I don't want to say too much about the area that you would also talk about, but people have tried to make predictions of the distribution of some infections with changes in climate. And I think the example you gave of dengue was a very good one, where people have already predicted that dengue would spread quite widely into Europe because of climate change. Fairly small changes in average temperature would favor spread of the distribution of the mosquito vector and would allow dengue to emerge in Europe. It's not always predictable is the other thing that I would say. So climate change will probably result in changes in land as well. And we don't quite know exactly what will happen, but that may actually remove breeding sites for some mosquitoes, particularly if standing water were to dry up more, there wouldn't be so much opportunity for them to breed. But certainly the temperature effect would favor quite a few pathogens. Do you want to say a little bit more, Colin, as well, particularly about the mosquitoes?
Yeah, one can elaborate a little bit, and there have been the crucial, along with the presence of a mosquito, it's also important to know if a disease is present in an area, and the word that's used is autochthonous. So if you've got autochthonous cases of a disorder, of a pathology somewhere, it means that it's spread from a resident of that area through a mosquito to another resident of that area. And there have been autochthonous cases of malaria in Greece recently, dengue in Italy and Sicily, and certainly the risks from that getting more prevalent and spreading northwards are high. And The area of Turkey and even up into Poland in terms of risks for dengue are high. And that in fact brings me back to a little question that might be worth pointing out from your presentation, is you had a hotspot of new disease up there in that part of Europe. And I think that climate change will certainly have quite significant effects on vectors and probably diseases in that area.
Yes, indeed. And I think that modeling study is very interesting for trying to predict these hotspots of disease emergence. But I think if you read carefully, the authors do mention that they don't think they were able to completely remove all of the confounding effect of reporting bias in their model. So some areas of Europe where there would be very good reporting may come out as looking like hotspots when actually it's only a consequence of the fact that they're good at detecting emerging infections rather than having all of the drivers for emergence as well. And I think it was just a bit difficult to dissect those things out right. in that study. But yeah, certainly I've heard others talk about, for example, the expectations of changes in West Nile virus. I know in the UK we seem to be seeing a gradual increase in Lyme disease, and that's probably due to temperature changes and things affecting the prevalence of the tick vectors for Lyme disease. So although it's very complicated, I think we do have much to worry about, that if we don't stop address climate change, we probably are going to see more and more pandemic threat or emerging infection threats, maybe not all pandemic threats.
Exactly. And here's an excellent question for you. Why was the SARS epidemic so much smaller than the SARS-CoV-2 pandemic, despite SARS having a higher R0?
Okay, so I think that's a good question. It's something I've thought about quite a bit, and my personal view is that it's probably because in SARS, most of the cases were very symptomatic and quite severe disease. So we were able to identify them more easily than with SARS-CoV-2, where initially the case definition was a severe pneumonia, but now we know that the spectrum of illness is much, much broader than that. And actually, we have asymptomatic individuals who almost certainly can transmit, but we don't see any symptoms in them. We have minimally symptomatic individuals, and then we have right up to very severe illness. So of course, if you can recognize the cases and you can act to isolate them, you can contain the spread of infection. But that strategy really didn't work. The UK tried to have a containment strategy at the start of this pandemic for SARS-CoV-2, and we were rapidly overwhelmed. So clearly we weren't managing to detect and contain all of the cases that were there.
Excellent. And there's a final question here. Has decreased air travel during COVID reduced the importation of tropical diseases into Europe? This is a question from our colleague, Terence Stevenson.
Okay, well, that's a great question from Terence. So I haven't seen data on this yet, but certainly our impression as an infectious disease service that we've seen fewer imported infections from what the regions you might consider to be tropical. So we have still had cases coming in, unfortunately, but I think fewer than we would normally be seeing. So yeah, that would be what you would expect if you had a big reduction in international air travel. I would just add a comment that I don't think it's going to be quite so simple in the long term that those will turn into sustained decreases, both because air travel will increase again, and also because the attention given to containing COVID-19 in many countries may have detracted from control efforts for other infections. It's not quite as bad as it could have been. There were predictions that this could have devastating consequences on malaria in much of Africa. But in fact, malaria control programs in many African countries were really organized and agile in their response and managed to continue their distribution of nets and achieve many of their targets well ahead of the pandemic causing problems in their countries. And actually the feared kind of resurgence of malaria probably isn't going to happen, but I suspect there will be knock-on effects, and we might actually see a bounce back with an increase in transmission of a variety of diseases in lower middle income countries, and then those being imported more frequently to the UK in years when our air travel gets back to normal, or into Europe when our air travel gets back to normal.
We do have a final question too, which is fascinating. It relates to a bit of zoology. Why are bats so often implicated as the animal reservoir of zoonotic infections? I'll start off, because there's one bit that I relate to mosquitoes and vectors, and that is that perhaps bats are considered sort of rare. But I was brought up in a part of the world where bats are incredibly common, and bats are small mammals, and they in Africa they were regarded largely as rodents, and rodents were everywhere and bats were everywhere. They're a really common small warm-blooded animal, and it's a double-edged sword. A lot of them eat mosquitoes, but a lot of them get bitten by mosquitoes, and they in fact are quite commonly, whenever you go into a cave full of bats, you'll find there's quite a lot of insects hovering around outside. And it's one of the ways of finding out where the bats are. So the mosquitoes will sometimes wait for bats in a way which seems kind of silly for a mosquito, but they certainly are a very good source for blood meals. And I think bats are prey to a lot of blood-sucking insects in the tropics. And as a consequence, they are exposed to a whole series of pathogens that are within that environment, spread to them partially by vectors.
Yeah, Colin, so I think you've hit the nail on the head, and that's also the understanding that I've come to more recently. So I used to think there must be something peculiar about bats that mean they're the sort of root of all evil. They seem to be at the origin of many of our emerging infection threats. But actually, my position on that has adjusted a bit recently, because there was a publication this year showing that actually, as you say, bats are incredibly common, but perhaps we just don't see them as much because of their nocturnal behaviors often. And also, they're incredibly diverse, and the diversity of viruses infecting bats is not really more than you would expect given the diversity of the species. So I think actually it's an issue of perception rather than that it's a reality that they are unusually the origin of more than you might expect. Yeah.
Well, the other point we didn't make, of course, is some of them live, not all bats, because some bats are pretty solitary creatures and live in small groups, but a lot of them live in incredibly complex colonies with vast numbers of individuals, so that they are a great place. It's like a crowded favela, if you like, inside a bat cave. There's a lot of animals in there, and sometimes you'll find several species within one cave. And essentially, I suspect that means that they are more adept at spreading viruses from one to the other.
Yeah, I'm sure you're right. I mean, I wish I could have changed my career and investigated bat viruses. Actually, it would have been really interesting.
Yeah, spend a lot of time in the tropics, I'm sure. Well, I think that's it. We don't seem to have any more questions here. So with that, I guess we could probably close the session. I think there's the final session probably opens in a quarter of an hour. But thank you very much for your presentation. I really enjoyed it, and it was very thorough and really very, very, very interesting.
And thanks to you, Colin, as well, for a brilliant summary of the mosquito and its contribution to human disease. So really enjoyed that. And I hope our audience have enjoyed the session too.